Hello, we are Lucy and Alyssa, and we're officially, unofficially, the best lab partnership in our AP Bio class.
For Unit 1, which is the chemistry of life, we know that life is made up of four main macromolecules. We got carbohydrates, lipids, proteins, and nucleic acids. And very crucial to this lab is the nucleic acids, since we're using DNA. We know that DNA is made up of an anti-parallel strand of nucleotides, which are nucleic acids, and they're bonded on a sugar phosphate backbone, and then additionally they're bonded between opposite nucleotides, which we can see in this figure here that shows the hydrogen bond between adenine and thymine and guanine and cytosine. We also can see the sugar phosphate backbone, but what we found most interesting about the bonds in this lab is that during electrophoresis, we have to denature the DNA, which separates it between these two nucleotides, so it separates that hydrogen bond that we can see in these two figures. And in the second figure, we can see that added heat, for example, denatures those bonds and then separates the strand.
Unit 2, Cell Structure and Function, is about cells and the different structures within them they have to help carry out different processes. The cell membrane has different proteins in it that help serve different functions. A type of breast cancer treatment is one that specifically targets plasma membrane receptor proteins in order to reduce side effects and decrease metastases and growth of the tumor. By knowing which alleles cause the cancer risk from the gel electrophoresis that we ran, we can better identify what proteins to target. The receptor proteins most commonly involved in the BRCA2 cancer are the receptor tyrosine kinases.
Unit 3 is cellular energetics, and we know that all of our cells are powered by ATP, which cells collect in a variety of ways, in a variety of steps. But in cellular respiration, the process is divided into glycolysis, the Krebs cycle, and the electron transport chain, two of which are within the mitochondria. We also know that a little bit of the ATP is made from glycolysis and Krebs, but most of it is made in the electron transport chain through a process called chemiosmosis, which is when the protons pass through ATP synthase and it physically attaches the third phosphate group to the ATP, which makes ATP. In this slide here, we can see the electron transport chain, and that is ATP synthase right there, going through chemiosmosis and attaching that phosphate group to ATP. We can see, though, that despite how efficient the electron transport chain is, cancer cells seem to prefer anabolic metabolism and glycolysis more than other steps of cellular respiration, even though they make less ATP, and they force these changes without signaling, unlike a normal cell would, making these cancer cells unable to use those signals to change the way that they metabolize could make them unable to replicate and grow, which is another way that we could target cancers like breast cancer. Unit 4 is cell cycle and cell communication, and the cell cycle is made up of G1, S, G2, and mitosis, and we know that through these phases there are certain checkpoints where the cell makes sure that it has certain things like the right amount of DNA, the right kind of materials to go through division, but cells that fail to pass some of these checkpoints might be sent to apoptosis, which is programmed cell death. So in this diagram, we can see that after the G2 checkpoint, a cell might be sent to apoptosis because it doesn't have the correct replicated DNA from the S phase. This prevents cells from replicating with miscopied DNA and mutations. All these checkpoints are monitored by cyclins and CDKs. They're very necessary to making sure that everything in a cell is okay before it moves on to the next stage. But a cell that didn't have these cyclins or CDKs or something else monitoring them could go through replication without regulation, which causes cells that might have a mutation or something wrong with them or not enough DNA to equally split to replicate, which would cause more problems, and then that would grow more, and that can lead to cancerous tumor growth. As we see in this diagram right here, the cell is on the G1 checkpoint and it has DNA damage, and that stops the cell and probably sends it into a resting stage or apoptosis because the CDK and cyclin bond together and form that complex. And a cell that couldn't do that would not be able to stop replication. We know that heredity is the passing off of traits/characters from parents to offspring, and those traits come from alleles that are created from the sequence of DNA found in a certain parent. Now, mutations in those sequences can lead to new alleles, and those may become more popular in a population, and that is what we see in the founder population of Ashkenazi Jews that have mutations in their BRCA1 and BRCA2 genes, which is reason for some breast cancers. We can see in this diagram that because the population of butterflies was mixed at first, and then a couple of those orange butterflies went off on their own, and a couple of the gray ones went off on their own, those new populations have less diverse organisms, which makes their gene pool smaller, which makes that trait more common, which is what happened with this mutation.
Unit 6, Gene Expression and Regulation, is the genes and their expressions that are passed on from parent to offspring. So the BRCA genes we're looking at are tumor suppressor genes that help aid in the repair of damaged DNA to prevent cancer. But when dysfunctional, the damaged DNA will divide out of control and cause cancer. Because Rachel and Deborah had the BRCA2 gene that caused them to develop breast cancer, their cell division breaks were defective, causing cancer to develop. So as you can see in the diagram, the healthy tissue has tumor suppressor genes with a working break, but the cancerous tissue has the tumor suppressor genes with a defective break. Unit 7, Natural Selection and Evolution. Natural selection is how a population's traits change over time to best fit their environment. And the population selects against people with the BRCA2 mutation because they will either not live long enough to reproduce, or their offspring will not live long enough to reproduce. The offspring of individuals who possess the BRCA2 mutation are more likely than normal individuals to express breast cancer, and these individuals are selected against. So as you can see in the diagram, some cancers are selected for over other cancers because they are able to live longer in the person. Unit 8, Ecology, is about biodiversity, community interactions, and how organisms respond to the environment. Different environmental factors have been found to play a part in the development of breast cancer or increasing that risk. And some of those things are environmental pollutants and toxic chemicals, and living too far from green spaces. By identifying these environmental factors that can contribute to the development of breast cancer, we can better understand why patients were diagnosed with breast cancer if they have no family history. Only about 10% of breast cancers are caused by the BRCA gene, and the other 90% can be caused by environmental factors.
Our first bioethical question is that if testing like this is common, then should people have to disclose with insurance companies the results they get from it? It's okay for insurance companies to know about medical history, but does that entitle them to results from predictive tests like these? Some may say yes. In the same way that insurance companies can know about previous driving infractions or history with car crashes and less medical examples like that, they should be able to know about a client's history in terms of medicine and predictions for their future. But some may say no because these predictions are not 100% accurate and are only based on genetics that might not 100% play out.
Our second bioethical question is whether or not people should use chemoprophylaxis, also known as chemoprevention. This is the preemptive use of medication as a preventative measure. So one position are the people who want to reduce their odds because you only have one life and you want to do all you can to stay healthy. But the other side of this is that some people think that it is unnatural because the person may never develop cancer in their life. They might just live with a high risk. And so they would go through this extensive treatment plan only to not have cancer develop. Also, this is very costly and not accessible to everyone, and it's not highly tested, so there are severe complications that come with it.